Hello everyone, my name is Gary O'Mealy, and in this video we are going to talk about atrophy and hypertrophy. So, the idea of kind of a lot of what we've been talking about with negative feedback regulation of endocrine glands and hormone secretion is that really, regardless of whether we're talking about hormone secretion by an endocrine gland, neurotransmitter released by a neuron, or any other kind of signaling mechanism that we've talked about up until now, cells in the body are going to find a way to adapt to how much signal they're being exposed to. Again, whether that's hormone or neurotransmitter, they're all signaling molecules, and cells have ways of adapting to what they're dealing with. So, as far as hormones go, the idea is very similar to what we've talked about in our very first lesson on homeostasis, all the way back at the beginning of the semester. So in this case, on the y-axis, we are looking at the serum concentration of pick a hormone, doesn't matter which one, and then on the x-axis, we're looking at time as hormone levels tend to kind of fluctuate a little bit. So similar to what we've looked at before, we have kind of a set point. It's not a hard and fast rule, but on average, this is where we want the hormone concentration to be. We have an upper limit. We don't want there to be more than this amount of hormone in the blood. And we have a lower limit. We don't want there to be any less than this amount. And then if we actually measure the hormone concentration throughout, say, a 24-hour window, it's going to look a little something like this. It fluctuates. Again, it doesn't have to be exactly the set point, but through negative feedback regulation, whether it's a simple endocrine reflex like what we're going to eventually see with the pancreas and insulin, or a more complicated mechanism like the hypothalamus, anterior pituitary, and thyroid, which again we'll look at in the future. The idea is whichever hormone in the chain we're talking about, we want it to fall within a normal range. The idea is that as we go throughout the day, when hormone levels start to rise a little bit and get kind of uncomfortably close to that upper limit, that's going to be where we want to shut off the hormone production. And then similarly, when hormone levels start to get a little too low, that's going to be where we want to initiate a mechanism for producing more of that hormone. So this is the reason why hormone levels in the blood fluctuate throughout the day, because there are times when hormone levels are low and we need to produce a little more. There are times when it's a little high and we need to kind of hold back on producing any more. So the idea here is that as long as we're doing a good job of this, there's not really anything to worry about, but you're probably at least somewhat aware that there are diseases in which hormonal secretion, hormonal regulation is not very well regulated. So there's all kinds of examples of this, and we'll talk about some of these examples in the future, like with diabetes, of course, and then also Graves' disease, Hashimoto's disease, which are both diseases of the thyroid, Cushing's disease, which is a disease of cortisol production. So the idea here is that if someone were to have one of those diseases in which it is characterized by poor regulation of hormonal secretion, that is going to inevitably lead to situations in which there is too much hormone or not enough hormone. And the purpose of this video is to show you that there are consequences for not regulating hormone levels properly, and those consequences can actually be seen and measured on the target tissues and on the target cells. So let's take a look at this. So hormones that affect the growth of the target cell are called trophic hormones, okay? So the best example of these are the hormones released by the anterior pituitary gland, okay? So that includes TSH, ACTH, growth hormone, you name it, okay? So the idea here is that if the body does a good job of regulating the secretion of these hormones so that the cells that are targets for these hormones receive adequate stimulation, those cells should not change in size over time. Keeping in mind the definition of trophic hormone is this is a hormone that causes the target cell to grow. If cells are starved of stimulation, meaning the target cell is not receiving adequate stimulation here, the hormone is undersecreted, we are not producing adequate amounts of this trophic hormone, this cell here is not ever going to be stimulated with this hormone, and over time, that is going to cause the cell to get smaller. We call that atrophy. On the other hand, if there is the opposite problem of there being way too much hormone in the bloodstream, way too much stimulation on your target cell, that is going to cause the cell to grow in size, and we call that hypertrophy. So, hormonal stimulation causes a target cell to produce a response. So, think of hypertrophy and atrophy here as being kind of the cell adapting to how much hormone is there. So think of the hypertrophy example first. The target cell is being bombarded with signal that is telling it to do whatever it does. So think of the green signal up here as instructions telling you to do X. So from the target cell's perspective here, there is a lot of message there telling it to do X. So by growing in size, the cell is attempting to increase its capability to keep up with the demand, okay? So for example, if a factory receives 100 orders a day to produce whatever product they're producing, we'll call them widgets, some kind of fictional product. If they receive 100 orders a day and they receive 100 orders a day for an entire year, the factory is going to adapt to that. They are going to work exactly as hard as it takes to produce 100 widgets a day. But if the next year they start getting orders for 1,000 widgets per day, how hard they were working and how capable they were the previous year is not going to cut it. They need to be 10 times more capable than they were. So it stands to reason that factory is going to have to get new machinery, hire new workers to make sure that they can keep up with the production. So this is why the cell is getting bigger. It is receiving a lot of orders here, and the cell grows in an attempt to keep up with the demand. On the other hand, atrophy is just the opposite. If the factory previously produced 100 widgets a day, and now suddenly it's only getting orders for 5 widgets a day, that's not going to cut it, right? So the factory is going to have to cut back, maybe sell some machinery to make up for the orders they're not getting, maybe lay some workers off, so the factory gets smaller because the demand is not there. So when it comes to hypertrophy and atrophy, you're probably most familiar with this in terms of the effect that these situations have on muscle mass. So weight training causes an increase in muscle mass because the skeletal muscles that we're working out as we lift weights, they become overstimulated with the neurotransmitter acetylcholine. Every time you flex a muscle, nerves from your brain are going down to that skeletal muscle and releasing acetylcholine onto it to get that muscle to contract. So from the muscle's perspective, the muscle, as it gets worked and worked and worked and worked, the muscle tries to keep up with that demand by synthesizing new contractile proteins and then adding them to the muscle, and over time that muscle starts to get bigger and bigger and bigger. On the opposite hand, if someone just sits on the couch and plays video games all day and never works out, never uses their muscles are going to atrophy because those muscles are hardly ever being stimulated with acetylcholine. So they get smaller and smaller over time, and they, of course, get less functional. So you've probably heard of hypertrophy and atrophy before when it comes to muscle, but the purpose of talking about this now is to tell you that atrophy and hypertrophy are not something that is unique to skeletal muscle. It is something that can be observed in any tissue that receives some type of signaling. So that includes not only skeletal muscle, but also cardiac muscle, and also smooth muscle, and also, of course, endocrine glands and tissues that are targets of hormonal reflexes. Okay? So if we look at a specific example here, consider the HPA axis, so hypothalamus pituitary axis, that we've already talked about before. So what we're going to see, and we'll see plenty of examples of these as we go throughout our next videos, if we do something to disrupt the complex regulation that's going on here, we may end up in a situation where a gland or a tissue is either overexposed to a particular hormone or underexposed. And based on what we've talked about, if we know that a tissue is over or underexposed, we should be able to predict what's going to happen to the size of that tissue over time, okay? So in a future lesson, when we talk about the thyroid, so I think that's just maybe 2 lessons away, we're going to see that this goiter that you see here, which is an enlarged thyroid, that is a thyroid that has gone through rapid hypertrophy over time, this is caused by a lack of proper regulation of thyroid hormone production, and this causes the thyroid to be overstimulated with TSH, which is one of those tropic hormones that comes from the anterior pituitary. So we'll talk a little bit more about how that comes to be in a couple of videos. So let's look at another example. Let's look at the HPA axis for the production of testosterone. You should hopefully be familiar with this by now. The hypothalamus senses a need for testosterone and produces GnRH. GnRH stimulates the anterior pituitary to produce both luteinizing hormone and follicle-stimulating hormone, which then stimulate the testes to produce testosterone. Testosterone circulates throughout the body and affects target tissues, leading to things like increased muscle mass, maintenance of the male reproductive system, including the prostate and the seminal vesicles. So as long as things are going right here, we shouldn't see a change in the size of anything here. So the idea, of course, is that when testosterone levels start to get a little low in the body, as I said, the hypothalamus senses a need for that, increases production of GnRH. That leads to an increase in LH and FSH, and then that causes the testes to produce more testosterone. Once we've produced enough testosterone, the negative feedback works here such that the high levels of testosterone reduce the levels of GnRH and LH and FSH, and that is eventually going to cause testosterone levels to go back down to normal. So as I said, as long as this negative feedback is working properly, the size of the tissues that you see here should not change much at all. But, hypothetical situation here, what if we inject a person with a super physiological dose of testosterone? The term super physiological here is meant to indicate a concentration of the hormone that exponentially exceeds what you would ever find naturally in someone's body, okay? So the typical concentration of a hormone is usually pretty low. Hormones tend to be pretty functional at low concentration, so we're talking at most in the micromolar to nanomolar range, usually more in kind of the femtomolar range, so that's 10 to the minus 12. But with a super physiological dose, we're talking something much, much, much greater than that, maybe like a millimolar dose, okay? So we're talking about injecting a person with testosterone, and there is more testosterone in this person's body than there ever has before by 1,000-fold, okay? So if this person keeps up these regular injections over time, this person is always going to be in a situation where there is a ton of testosterone in their body. Well, what that's going to cause is that's going to cause the negative feedback here to always be active, so that because testosterone levels are so high, the hypothalamus is never producing GnRH, and similarly the anterior pituitary is never producing luteinizing hormone or follicle-stimulating hormone. As a consequence, the anterior pituitary is understimulated because there's no GnRH, and it will start to atrophy. The testes, because there is no LH or FSH, similarly will start to atrophy. They get smaller because they're not being stimulated with anything. But because there is so much testosterone, all the tissues that are targets of testosterone, like skeletal muscle, the prostate, the seminal vesicles, they are being absolutely bombarded with testosterone 24 hours a day. So to keep up with demand, they start growing and getting bigger. And this is something that we'll talk about in the future when we talk about gonadal hormones, and we'll look at the specific example of the steroid scandal in baseball from 20 years ago. But the whole idea of abusing anabolic steroids in something like baseball or some sort of competitive sporting event is that if you inject yourself with testosterone, you build muscle mass, you get really strong so that you can hit a lot of home runs and make a lot of money. But the problem is that, and we'll talk about this in that video, the problem is what you can't see. You can't really tell that the testes are getting smaller, or especially that your anterior pituitary is getting smaller. So external injection of hormones is a tricky business to get into. You don't want to inject too much such that you screw up the natural regulation of things. Okay, that's going to do it for this video. Here is a list of vocabulary terms you probably want to be aware of. And for checking your understanding, number 1, how can the comparative sizes of endocrine tissues compared to a normal size be used as a measure of whether hormone production is normal or not? So say you have an understanding that the size of the thyroid is typically like, we'll say 100 grams. Okay, that's just kind of an example. So if the thyroid is supposed to be about 100 grams, and then you talk to someone and you have some way of measuring the size of their thyroid, and their thyroid is 25 grams, what does that tell you? What does that tell you about how much TSH there is, how much thyroid hormone there is? That's kind of what I want you to think about here. And thinking about this question is going to be really important for the lab that you're going to do this week. And lastly, if we were to inject a person with a large amount of an unknown tropic hormone, could we identify the hormone based on how the tissue sizes change? So answering a question like this is going to require 2 things. Number 1, you have to know what that hormone is and what its targets are. So if you know the targets for each hormone, you can pay attention to how things change and make a prediction as to what hormone you're dealing with. Secondly, you need to know how hormonal negative feedback regulation works, and we obviously talked about that in the previous video. Okay, that's going to do it. If you have any questions, go ahead and drop them in the comments section, and I will see you next time. So long.